first up is going to be Dr. Andy Tay, or is it professor now? You're now officially a professor. So Andy is currently muted. He'll be on in a second. Andy has an outstanding CV, as does Elina, who being a Forbes 30 under 30. I don't know if anyone knows about the 30 under 30 program. It's very prestigious, very important position or award to be awarded. Was the 1851 Royal Commission Brunel Research Fellow at Imperial for a few years, and very recently, in fact, starting this year, I believe, is returning to NUS Singapore to start his own lab, and we wish him all the very best in that. But before then, we've just had an opportunity to grab him before he goes. Well, kind of, he's still in Singapore, so ignore the yawning. And we'll look forward to what he has to say as a sort of farewell from the department. So Andy, without further ado, please take it away.
Thank you, Chris. Let me try to share my screen. Share screen. Okay. Can you see the slides?
Just a few seconds. Yep, now we got them.
Okay, cool. So hello everybody. I'm Andy. So good afternoon if you're coming from Europe, London, and wherever you are from the rest of the world. So I thought that it would be interesting for me to give this talk and to think about how our experience during the COVID-19 pandemic can be actually made related to my research. And that is why I decided to have this title, How Lessons from Online Shopping Can Benefit Cancer Therapy, because I believe that during the COVID lockdown, most of us have actually experienced online shopping. So a bit about myself. I completed my bachelor's degree in the National University of Singapore, and then I went on to UCLA for my PhD, and I did my postdoc at Stanford and Imperial College London. So my lab will be, because of COVID, there's a delay, but the physical lab will be ready early next year. So I wanted to start by asking a question whether anybody has done online food shopping before during the lockdown. So during the lockdown, I was back in Singapore, and these are some of the food that I ordered. So if there's any Singaporean audience, I think you may be familiar with some of the food. So there's chocolate cake from Awfully Chocolate, sushi, bubble tea, and also Shake Shack, but not from Singapore, but America. And during this lockdown, I was ordering food through these different delivery platforms, and these are the common food delivery services we have in Singapore. And through this process, I realized that in fact there are many factors to consider when we are trying to choose what type of food delivery service that we want. Is the service going to be as efficient as I think? What is it going to cost me in terms of money? It is very common for people to open 3 different apps together and compare whether there's promo code or any discounts. So cost is definitely an important factor. And also very importantly, whether it causes you any emotional stresses. I remember ordering sushi during the lockdown, and what happened was the delivery man was late for 3 hours, so the sushi was not edible, and it came only at like 10 p.m. So I was pretty angry about it. And the reason why I'm talking so much about food delivery services is to draw an analogy about the importance of delivering DNA to cancer treatment in a form of an emerging therapy known as cancer immunotherapy. So now I'd like to start the presentation more proper and looking into the questions of cancer and cancer immunotherapy. So we all know that cancer is a very deadly disease. In fact, 1 in 3 to 4 people globally is at risk of developing cancer over their lifetime. And since the 2015, cancer has accounted for more than 9 million deaths each year, and it currently accounts for about 30% of death in the world. And cancer cases have been increasing each year due to a variety of factors like pollution, diet as well. And throughout all these years, there have been amazing medical advances, but I think none of them is as promising or as groundbreaking as a type of cell-based cancer immunotherapy known as CAR T-cell therapy. So this is how CAR T cells work. You imagine there is a type of cell in your body. These are immune cells known as T cells. They are soldiers that patrol around your body looking for the enemy cells, which in this case is our cancer cells. And when these T cells encounter the enemy cancer cells, they will try to destroy it. And that is basically what cell-based cancer immunotherapy about, using T cells' protective defensive function to destroy the cancer cells. And this is how CAR T cell therapy work. Firstly, blood is being isolated from the patients and T cells are being isolated from the blood. Next, through a process known as transfection, the DNA will be introduced into the T cells to genetically engineer them into CAR T cells. And the DNA encoding for this CAR gene, you can think of them as proteins that actually make the T cells stronger or better in terms of recognizing the cancer cells. Next, these CAR T cells are being grown into large numbers before being reinfused into patients where they can attack the cancer cells. And basically, this is really what CAR T cells is all about. We have a T cells, and we need to introduce DNA into the T cells to engineer them into CAR T cells, which is like a super soldier. And this process of delivering DNA is known as transfection. And the process of transfection can take place mainly through 4 different stages. The first step is packaging of the cargo. It will be packaging of cargoes like oligonucleotides like RNA, DNA, or proteins, depending on the application. Next, the cargo has to cross the cell membrane of the T cell, and they can do so through different mechanisms such as endocytosis, poration, or penetration. Once inside the intracellular spaces or the cytoplasm, what happens is that this cargo has to unpack for them to be released into these intracellular spaces. And if the intended purpose is for applications such as genome integration or gene editing, the cargo also has to be delivered into the nucleus. And so this is a transfection analogy I have built for some of you who may be new to the concept of transfection. So the first step of cargo packing is very similar to you ordering your goods, and then these goods have to be packaged in a factory. And the step 2 of transmembrane transport of cargo across the cell membrane is really analogous to goods being delivered to your house. And you can imagine the gates or your doors in your apartments or in your houses are similar to the cell membrane. In the third step, this is where you unpack the goods, and that is similar to unpacking of the cargo in the intracellular spaces. And lastly, after you unpack the good, you will use the goods. And in this case, it will be analogous or similar to the goods being transported into the nucleus for application like gene editing, which is important for CAR T cell therapy. There are in fact actually a variety of methods people have been using to deliver DNA into T cells to engineer them into CAR T cells. So I'll talk about a few of them, and then I'll go and introduce how I'm using nanotechnology to develop a method which I think is actually more efficient and also less perturbative to cells. So the most popular method is using viral vectors. And viral vector is popular because clinicians have a long history of using them. And usually many companies have set up large-scale manufacturing factories to produce these viral vectors. But viral vectors have 2 main limitations. The first is immunogenicity. What happens is that most viral vectors do integrate the genetic elements into the host genome. And what happens is that when these viral DNA are being transcribed and translated, and these viral proteins are being expressed, it can elicit immune responses. And in fact, in many gene therapy trials, there have been very adverse immunogenicity reaction leading to deaths of patients. But so far, there hasn't been any similar cases in CAR T cells, but we shouldn't rule that out. And a good analogy of using viral vector is sort of like a food delivery with variable levels of poison, which means that the T cells is actually slowly incorporating this DNA from the virus. And we do not know whether at what point would there be a very adverse immune response because of a large number of viral proteins being expressed. Another danger is that when viruses are introducing the gene into the host genome, they do it randomly. And what happens is that when these genes are being inserted near hyperactive promoters or proto-oncogenic sites, it can enhance the risk of cancer. So it's pretty ironic that you are trying to solve cancer with CAR T cells, but your delivery method may actually put the patient at risk of developing cancer after the treatment. And from an engineering point of view, the main limitation of viral vector is that they have a very low cargo load. So if you look at, so this is Grab. Grab is similar to Uber, but it's mostly based in Southeast Asia. So when you are ordering food from this delivery platform, what happens is that you may get delivery men cycling on a motorbike or in a car. And similar to viruses, from these pictures, you understand that different vehicles actually have a limitation of how much goods or how much food they can deliver. For most viral vector, the maximum cargo load is about 5 to 15 kilo base pair. And so if you are thinking of engineering a more complex cells with a larger protein or more complex biological functions, viral vector may not be the ideal transfection agent. And also importantly, viruses can only deliver DNA or RNA, and therefore for some gene application such as CRISPR, where you may require a guide RNA or Cas9 protein, this may not be the best method that you'll go for. So as a result of this limitation of viral vector, for the past 20 to 30 years, there has been a lot of interest to look for non-viral, cargo agnostic kind of method. And being cargo agnostic, I mean that the method doesn't discriminate against the different type of cargo species. They can deliver anything as long as the size is appropriate. And bulk electroporation has really been the technique that has been used by cancer immunologists for CAR T cell therapy as a non-viral kind of alternative. What bulk electroporation does is that basically we are subjecting cells to very strong electric fields, usually about a few hundred volts, and it creates a streaming potential that wraps around the cell and pops the membrane to create pores of different sizes and also different length that they will be staying open. And what happened is that with the cargo at the extracellular media or the spaces outside the cells, they will then rush into the cells, either through diffusion or random probabilistic entry into the cells. But the high use of voltage actually leads to Joule heating, which has led to very low cell viability when using bulk electroporation. And also, we recently showed that bulk electroporation is a quite intense process, and it also affects cell proliferation and cytokine expression. So basically, it is not very healthy for the cells to undergo bulk electroporation. And one good analogy is a food delivery that destroys a home. Imagine ordering a sushi, and you realize that in order for that delivery man or woman to deliver sushi into your home, the person has to tear down your gate or remove some bricks from your house. And I can imagine that would be a very stressful experience, and likewise for cells as well. An emerging technique is the use of nanoparticles, either through lipid nanoparticles that encapsulate the cargo, or gold or magnetic iron oxide nanoparticles that are being conjugated to the cargo. But a main limitation of nanoparticle is that immune cells do not readily take up this nanoparticle except for phagocytic macrophages. And another problem is that once these nanoparticles are being endocytosized within the cells, they are being trapped in what we call an endosome. And the problem with endosome is that there is, even after so many years, there's really no good solution to control when the cargo is being released from the endosome. So this is analogous to a food delivery with no estimated time to arrive. And you can imagine how frustrating it can be, and also how inconsistent the performance can be. Another emerging method is the use of microfluidics. It is an advantage because microfluidic platform can operate on a continuous flow or in a parallel format, and therefore they can do a very high throughput transfection of the cells. But the main problem is that microfluidic is quite prone to clogging. So when clogging happens in any of these microchannel, it actually disrupts the continuous flow. And if you look at figure B, in order for this cargo to enter the cells, it means that the cargo must be present in the entire microchannel. And for some cargo which are more rare or more expensive to produce, this can lead to a significant cost of production for transfection. And I think also what is interesting is that many of the patients who undergo CAR T cell therapy, they are actually at the late stage of their cancer. So there are very, very low number of fit or T cells that we can isolate from these patients with late stage cancer. So it is also worthwhile to think about whether there is a need for us to go to extremely high throughput using microfluidic as well. So in my research, we have developed high aspect ratio nanostructures for this kind of like T cell transfection. So in nano, I mean they are about 10 to the power minus 9 meters. And it is important to stress that they are not the same as the nanoparticles because they are high aspect ratio. They are also not freely floating, and they use quite different transfection mechanism. So for nanoparticles, it's mostly using endocytosis, whereas for these high aspect ratio nanostructures, they use a variety of mechanisms, including transfection, poration, electric field, and even magnetic field. And this technology is really possible because of the electronics industry, and we are really borrowing techniques from the electronics fabrication industry to make this kind of high aspect ratio nanostructures for T cell transfection. So with a nanofabrication technique, we can actually produce what we call a hollow nanoshells, like you can see in this scanning electron microscopy images. And this is the process how we actually perform transfection. And we actually name this method magnetic nanoelectroinjection, and I will explain why. So the first step is that we will label the T cells with magnetic particle, and this is just part of the T cell manufacturing process. Is routinely done to use magnetic particles to activate the T cells. And with a magnetic force, we actually pull and localize the T cells very near to these hollow nanostructures that we have fabricated. Next, we apply electric field. And because only the nanochannels are filled with fluids, and therefore they offer the path of least electrical resistance. And therefore, because of that, we can apply very localized electroporation through each nano channel to open the pores on a cell membrane for cargo entry. And also because DNA is negatively charged, so as we apply a positive charge at the top of the cell, we can also provide an additional electric force in addition to diffusion gradient to inject the DNA into the T cells. And finally, we apply magnetic force to bias transport into nuclei by altering kinesin expression. But due to time consideration, I will not be talking so much into the biological mechanism. But as you can see, because we are integrating techniques from the nanoscale fabrication and electrical and magnetic modalities, that is why we decided to name this technology a magnetic nanoelectroinjection, or MAC-NEI for short. So with this technology, we actually compare it against the gold standard virus. In this case, we use a lentivirus instead of adeno-associated viruses. We also compare against a Lonza bioelectroporation, which is FDA approved for CAR T cell therapy. We showed that our technique was able to offer about 50% net transfection efficiency with about a 2-week stable gene expression. And that is actually considering quite good, about 3 to 4 times better. We also compare our technology against a fresh or cryopreserved T cells, and the performance was quite similar as well. And it is important to stress that just 2 years ago, there was a paper published in Nature where they were able to get about 22% transfection efficiency. And I think this really highlights that there's a strong need for us to, from a biomedical engineering point of view, to combine our engineering methods and work with cancer immunologists to try to improve transfection of T cells for cancer immunotherapy applications. But throughout all this research, we also realized that it is not just a transfection efficiency that matters, but also what is the cell quality of the transfection. What we mean is that you can get a high number of transfected cells which are expressing the CAR gene that you want. But if these cells are not very healthy, they do not grow well, and when they are introduced in the body, they do not migrate to the solid tumors, the cancer treatment will not be efficient. So we set out to design a series of tests to look at and to assess the cell quality after transfection. The first thing that we did was to look at calcium stresses. So calcium are normally maintained at very low intracellular level, as you can see in the control when we use a fluorescent calcium indicator. So when the cells are stressed, such as after being transfected with a viral vector, you can see that there's an enhanced or elevated intracellular calcium level. So these are results presented 24 hours after transfection. You can see that with virus and bulk electroporation, there was significant increase in calcium stress. And whereas for MAC-NEI, there was actually quite minimal calcium stress. We also look at whether the cells are able to proliferate as well. And that is important because it actually affects cost. The cost of producing these cells actually directly impacts how much the CAR T-cell therapy will cost. So we actually monitored cell proliferation over 2 weeks, which is a typical time that industry uses to produce sufficient cells for treating and injecting these cells into patients. So in general, we found that virus and bulk electroporated transfected T cells proliferated about 20 to 30% times slower. And this is actually quite important because nobody has really looked into this, and because there was no awareness that transfection could have such a long and also adverse downstream implication. Currently, CAR T cell therapy costs about $500,000, and the bulk of the cost is actually attributed to the cell manufacturing expansion process. So the goal is that if we can develop a transfection method that's minimally perturbative to the cell proliferation, we could potentially reduce the cost of the treatment, but also reduce the delay in terms of patients getting the cells for their treatment. We also look at whether the cells were able to migrate normally. So this is a very simple transwell assay where we actually look at how these transfected T cells responds to chemoattractant. And the reason that we do so is that because some of these T cells, we want them to traffic or to migrate to a solid tumor for solid tumor treatment. So in general, we found that bulk electroporation and virus-treated T cells, they generally migrated about up to 80% times slower than those transfected with MAC-NEI. We also look at RNA expression, and that there was actually a least change in gene expression with MAC-NEI as well, which is actually quite important because we do not want cells to be so stressed that they actually modify their protein synthesis by a lot. So in summary, we have shown that we are able to integrate fabrication technology from the electronics industry to create these high aspect ratio nanostructures to deliver DNA with quite high efficiency, I would say. And also we have shown through a series of in vitro tests that MAC-NEI actually causes minimal cell perturbation. But I also thought that because there are graduate students, postdocs, or even undergrads who may be attending this seminar, so I thought that it would be quite nice to share a bit about these different career options. So definitely, I think one way is academic, because there's a lot of interest in the academic area to look at CAR T cells for cancer treatment. And it is not just in the basic cancer or immunologist area, but also a lot in terms of the engineering area, so that we can translate technology from the bench to the bedside. Another area that's actually extremely promising is going to the industry. The CAR T cells industry is expected to explode and tremendously, and it's promising because it means that we will be able to, in the future, be able to use cells as living medicine. And there's actually a lot of job opportunities in startups and even big pharma companies like Novartis and Kite Pharma as well. There's also a lot of opportunities career in terms of consulting. That means, for example, like startup or companies trying to move from an area that they have already done or a country that they've already done and moving to other countries. For example, moving from the United States to Singapore, and we actually need consultant with good knowledge of the domestic market. So in terms of that would also apply to the United Kingdom as well. Lastly, there will also be jobs in terms of science writing. So for example, recently I wrote a freelance article on non-viral genetic engineering of CAR T cells. So I hope that through this presentation, you actually learn that this is a promising field and there are many career options. So if you're interested to go into research or industry or into writing, it is good for you to actually be more familiar with this area. So this is also my website with a QR code. So immune cell engineering is one aspect that I'll be doing, but there will be two other areas. So if you're interested to learn more about my research, you can scan this QR code, and then if you have any questions, we can discuss during the Q&A. Thank you.
Fantastic. So on behalf of everyone, I'm going to offer you a round of applause. I'm going to take the place of everyone else on the chat and do so. First of all, thank you for a fantastic talk. We do actually have a question already for you, so if you wouldn't mind. Naomi Nakayama has asked, Andy, your nanosurface has interestingly patterned needles. Does this pattern matter?
So in this case, we are not really using like needles, but because if I would think of needles, I would think of needles with a certain sharpness. They are quite sharp, but these are really like straws. You can think of if you are trying to drink a beverage, you're using a straw, and these are really like hollow straws. And in our case, we do find that there are some importance of having certain patterns, but not so much of the pattern per se, but more of like the density of the straws. For example, if you have a more dense straws, it means that there will be more straws contacting the T cells. So there's actually more probability that we can apply electroporation to open the cell membrane for DNA to be injected into the T cells. Yeah, and also we have looked at other parameters like whether the height and the diameter of the straws affect transfection. There is some sort of connection, but I wouldn't say that they affect the transfection performance as much as the density of the straws.
Okay, that's great. Thanks very much. I just wanted to point out to everyone that's listening, if you do want to ask a question, we still have time for questions. If you'd like to either put your hand up and you can ask live, or if you don't want to do that, you can put a question in the chat. Like Jun Ishihara, who asks, have you ever looked at what type of T cells are effectively transformed, like the CD4 plus or the CD8 plus, and does that make a difference?
Yeah. So right now, all the data that I presented is just like T cells in general, but we know that actually the T cell population is quite diverse. So there is a helper T cells like CD4 plus or cytotoxic CD8 positive T cells. So I have not presented data here, but we have shown that they actually do work for CD4 plus and CD8 plus quite uniformly. Slightly lower for CD4 plus, which we don't really understand. But what I think is actually more interesting is looking at how this transfection performance is different in these T cells from patients of different age groups. So what we have been trying to do, what I'm planning to do is to actually get T cells from cancer patients of different ages, and then looking at how, using the same parameter, how would the transfection performance be different? Yeah, and that is actually quite important because if you look at most of the published literature, most of the papers are reporting using T cells either from healthy controls or they are from cancer patients, which are quite young. But in fact, actually, most of the people who are going for cancer immunotherapy, they are really quite old and they are in a late stage cancer. So I think that there's actually a huge need for us to look at how the transfection performance actually differ across immune cells of different ages, so that maybe from this raw data, we can sort of build a model for us to understand how changes in certain biophysical parameters of the cell, such as whether they are more permeable to electric fields, how would their cytoskeleton stiffness affect DNA transport within the cell, and all that, yeah, to look at transfection performance.
Fantastic. Thank you very much. We have time for one more question. So particularly if there's students or early stage academics that feel like they would like to ask a question, now's the time. Well, I'll ask a very quick one whilst people are coming up with their last questions. You compared against bulk electroporation and viral transfection. Was there a reason why you didn't compare against, say, chemical techniques like lipofectamine or Viafect, I think it's called, or what other alternatives?
Yeah, lipofectamine is one of the most popular one. So lipofectamine works quite well for a lymphocyte cell line called Jurkat cells. And in fact, we've actually have a paper to compare across for lipofectamine for Jurkat cells as well. They were okay, but they are quite stressful to the cells. And for T cells, lipofectamine is really not the gold standard that people are using. So when we compare against virus and boiled electroporation, we are looking at FDA-approved gold standard methods to compare. Yeah. So another limitation of this lipofectamine is that because we are already introducing an external exogenous DNA into the cell. So if you think of lipofectamine as a material, you wouldn't want to introduce things that that's in additional to the DNA that you're trying to deliver. And we are not sure, what are these excess lipofectamine going to do to the cells once they're inside. So another advantage that I'd like to highlight with this kind of high aspect nanostructure technology is that we are only delivering the things that you want. So, unlike a virus vector, we do not have the virus genes. Unlike a nanoparticle, we do not introduce any nanoparticle material inside the cell. Yeah, so that is actually an advantage because we only deliver what the clinicians want us to deliver into the cells.
Fantastic. And we do have one last question, not on the science so much, but you were very kind in mentioning the various job opportunities and stuff available. Would you have any recommendations or advice for early career researchers who are looking to kind of follow in your footsteps, as it were?
Right. So I will consider myself as an early career researcher as well. So I'm not sure whether what stage of career you're in, but I think that there's actually a lot of promising jobs in terms of post, whether they are in the academic lab or even in the industry. There are many industries which I mentioned, like Novartis, Kite Pharma. So these are the big pharma companies that already have FDA-approved CAR T cell. But if you look at the number of clinical trials on CAR T cells, there are more than 500 alone in the world. And I believe that there are many trials going to happen in the UK as well. So I think that whether is it an industry or academic lab, there will be lots of career opportunities. But I think that this field is also quite diverse because they are not just looking at immunologists, cancer biologists, but they are also interested to find engineers who are able to develop transfection technique, which I mentioned. But also, what about things like large scale bioreactor, so that we can grow these T cells to large numbers. So and I think that one of the best way to really look into all these opportunities is to read up a lot. So there are many free online magazines like Gen News, The Scientist, or even and you can look at them and look at how industries are approaching this problem. So if you're interested to join industry, you sort of have an idea what are the problems they are facing, and then when you go for interview, you are able to pitch to them what has your education taught you that you're able to solve their problems. Yeah.
Fantastic. That's absolutely brilliant. Thank you very much for your time. I'm afraid we're going to have to cut you short because we're running out of time. But thank you very much, particularly for staying up so late because we're not in Singapore. We don't appreciate it. But yeah, it's been a pleasure. Thank you.
Thank you, Chris. Right. Thanks for having me.
So we shall move on very swiftly to ELINE van der Kruk, who joins us from the Netherlands. She is also a former fellow in the department and has very successfully transitioned to an academic career in the Netherlands. She calls herself the first sports engineer in the Netherlands, which I think is an excellent title, and has worked with such luminary people as the speed skating team of the Netherlands and won an NWA Rubicon grant to join us a few years ago in Imperial. And we wish her very well in her new position in TU Delft. But in the meantime, we're looking forward to hearing what she has to say about her research to date.
I was still muted. That wasn't really helpful. So thank you. I'm really glad that I am invited to give a talk today about human movements because I'm really fascinated about human movements, and particularly I'm fascinated about the boundaries of human movements. So when you think about the boundaries of human movement, of course, you very quickly think about sports. see if I can get the next slide. There you go. Because in sports, that what we're trying to reach there is optimal performance. So that's one side of the boundaries, but that's not the whole story. When I think about the boundaries of human movement, I think about two boundaries. On one side, you have optimal performance in sports, and on the other side, you also have movement limitations. So, for instance, when we age, or it could also be due to a neuromuscular disease, or even an injury in sport, that means that we're hitting the lower boundaries. So my research really focuses on pushing the boundaries of human movements. And working on the boundaries also means that you're always working on the edges. So that means that if we want to look into elite athletes, if we want to do that doing experiments or tests, you usually only have N is 1, because there's only one athlete that can go as fast, like that can actually do the fastest trial in something. On the other hand, if you want to look into the lower boundaries, so the movement limitations, it's kind of difficult to look into someone getting injured. It's not really ethical to get someone injured during an experiment. And that's also why my research is closely connected to predictive modeling. So we really work with predictive models, translating humans into physics. And that could be a rigid body model, but it could also be a neuromuscular model. But once we have a model of our movements, we can use that to simulate the movements, and we can also use that to optimize the movement. So what I want to talk about today, so the title of my talk is What Moves Us? And in this talk, I don't want to answer that question, but I want to take you on a journey that I took to actually come to this question. And for that, I'm going to take you to these upper boundaries, so to this optimal performance in speed skating. And I'm also going to take you to the lower boundaries, so to the movement limitations. But let's start off. I mean, we're exactly 1 month before Christmas. So let's start off with speed skating. So maybe at some point in your life you did some ice skating, and it could be that that the first time you tried this, you actually fell over, because speed skating is ice skating or speed skating is quite a difficult movement, and it's very different from the locomotion that we usually do in daily life. So if you walk, you usually push off backwards. So that means you're pushing off in a direction that you want to go to. Force is mass times acceleration. So you're generating a force in a direction where you want to get your acceleration. This is something that's impossible to do on ice skates, because you're standing on a very thin blade and also on a very slippery floor. So if you want trying to push off backwards, like in walking, you will slip away and you will fall over. So the only way for you to generate a forward motion is by steering your skate. And if you're really good in speed skating, the way to do that is to make a curve with your skate. And throughout this curve, throughout the steering, you're trying to push off. And that way, you're not only pushing off forwards, but you also generate a force sideways. So you also partly move to the sides. And that's why when you're trying to skate, you will always have a movement that goes in both directions. So you always move from left to right to get yourself forward. Well, I'm not a really good skater myself. Let's start out with that. But I am really fascinated by this movement. So skating is the second fastest way for a human to propel themselves over the flat land. The fastest way is actually cycling. So if you think about, well, if I think back, about this ice rink in South Ken, I can assume that you're not going really fast on that ice rink, and I'm not really going fast when I speed skate. So I brought a video from the team that I've worked with in this project, just to give you an idea of what this speed looks like. So these skaters actually reach velocities up to 50 to 60 kilometers an hour. Just a second, I can't see the screens anymore. Yeah, I'm back, sorry. So they reach velocities of 50 to 60 kilometers an hour. And there's another thing very fascinating about skating, because if we look at the skating techniques, we see that there are a lot of different techniques, and even the Olympic skaters and the world champions use these different techniques. So I brought a video of one skater imitating different skating techniques that have been used by either Olympic or world champions over the years. So it seems like there are a lot of different techniques, and there are also a lot of different speed skaters, actually. The speed skaters differ a lot in body builds. So it seemed like there was an optimal technique for an individual speed skater. And that is exactly why we wanted to find out what the optimal technique was. And one of the reasons why we started to make predictive models. And we built a model of a speed skater, and we called this the simplest skater model. Because it really is a very simple model. The skater only consists of two masses. So one mass at the skate, and the skate can steer, so it can make this actual curve to curve over the ice, and a mass at the upper body. And the changing distance between these two masses is actually your leg extension. So that's how much you push off. And by steering your skate, by having this steering angle in the skate in the model, we could simulate how different steering angles eventually result in different techniques of bringing you over the ice. And of course, it's very nice that we had this very simple model, but we had to convince people that this is actually what a speed skater does. So we had to do a validation study. And usually when you want to validate a model, especially in human movement, you go to a human movement motion capture lab, where we can measure the movement of a person in 3D. And we can also capture their push-up forces, so the ground reaction forces. Unfortunately, I couldn't really bring ice to the lab, and I also couldn't really bring force plates onto an ice rink. So we had to come up with a clever way of doing this, and we built several instrumented speed skates. So basically, we made a mobile force plates, and this is one of these skates. And this is what we so-call a clap skate. That's what they usually skate on. So there's a hinge between the blade and the shoe. And in between, you see a bridge, and that bridge is where the force sensors are. So we could measure the push-up forces of the skaters in 3 directions, and the skater used their own shoe and their own blades. The data were collected on a data logger, and this data was then sent to a phone. So we could collect the data, but also from the phone, we could send it to a Google Glass so that skaters could see real time what they were doing on the ice. The other thing was that we also had to capture them in 3D, because that's also what we do usually in the lab with a motion capture system. But in the lab, that's about 8, you use a volume that's about 8 meters long. But one speed skating stroke was already 12 meters. And because we wanted to capture 4 strokes, we had to had a huge volume. So we brought the system to the ice rink and captured what at that point was one of the biggest volumes. At the moment, I think they've captured bigger volumes than this, but this is what the eventual measurements look like. So with these data from the motion capture system, so we had 3D kinematics and we had the force data, and we could verify the simple skater model. And this very simple model turned out to be able to simulate the speed skating movements in less than 3% error in both position and velocity. So that's very nice, because now we have a model in which we could look at the timing of when off in the curve of your skates, how fast to move away, so how fast to push off. Basically, that's the changing distance between this upper body and your skates. And you could optimize the skating motion depending on, for instance, how fast you're able to push off. You could change the mass distribution. You could change the length of someone's legs. So you could look into different strategies for different skaters. What you need to do for this is you need to have a movement objective. So you need to know what am I going to optimize for? And it seems clear that within skating, you want to go as fast as possible. But what if this skater has to do this skating stroke as fast as possible multiple times? What if you want to go 10 kilometers instead of 500 meters? Then suddenly this movement objective is not just velocity anymore. And that brings us closer to the question, what moves us? And this brings me also to the next part in this talk, because I've talked about the the optimal performance now, this upper boundary. But I'm also intrigued by these lower boundaries, by the movement limitations. And that's what we've worked on in London in the past 2 years. So in these lower boundaries, well, let me start with a little bit of sad news. So this is about age-related decline, and we're looking into how age-related decline eventually results in movement limitations. And I think, except for a couple of freshmen that are maybe in a team call right now, every single one of us already started on our age-related decline. Because that starts around your 20s. It's different for the different systems in our body. So the onset in our neural system is earlier than the onset in our muscular system or skeletal system. But yeah, it started in our 20s and it goes very slowly in decline from there. Of course, it's also different for different individuals. Hopefully, probably you haven't noticed much of this yet. And that has to do with the fact that we are, as humans, we're really redundant. And that's in two ways. So first of all, we are redundant because we have way more muscles than necessary to make one particular movement. So we could do the same movement using a different muscle recruitment. On the other hand, we're also redundant in the sense that, for instance, for daily life activities, we have more capacity than needed. So before we hit the boundary of not being able to perform a certain task, we still have quite some reserve. And if we do happen to get to not be able to make a certain movement anymore, so we can change our muscle recruitment, and that could also be changing our movements. So for instance, if you want to get out of your chair now and you're struggling with this, you will start changing the way you move your body. So that could be either changing the way you move your upper body. You could start using pushing off on your thighs, or you could even start pushing off on your armrest to get out of your chair. So this interplay between capacity and compensation is something we got interested in, and it's something that we wanted to test in an experiment. And therefore, we set up an experiment in London where we measured both capacity and compensation. So we measured 50 participants, half of them elderly, half of them young. And we did a lot of measurements in capacity. So we measured strength, we measured nerve conduction, we looked at their balance, we did some proprioceptive testing. So looking at basically the sense you have in your joints. We asked them about their lifestyles, and we did a cognitive test as well. And on the other hand, we looked at compensation. So we observed them doing one particular task, namely standing up from a chair and walking to a table. And we observed them and also measure them. So we captured the whole 3D kinematics again, and also the push-up forces. We asked them to do that 5 times without giving any instructions. They were sitting on a chair and we asked them to walk through a table as if they would pick up a phone. Afterwards, the second time, we asked them to do that as fast as possible. And then finally, we asked them to stand up with using different strategies, the strategies you just saw. So standing up, pushing off in your armrest, pushing off in your thighs, or not pushing off at all. So this is a huge experiment. We're still looking into a lot of this. So a lot of the results aren't clear to us yet. But I want to pick out one element from this, because looking into the previous literature, we saw that what basically is expected is that you start to compensate because your capacity, or mainly what's been looked at is strength, is going down. And if you don't have enough strength, you need to start to change your movements. Well, if we look at our results, and we look at these strength measures and the compensation strategies, we actually see that, well, yes, the elderly men had less strength than the young men, which is totally expected because they have an age-related decline. But in absolute sense, if you compare the young women to the elderly men, actually, in absolute sense, their strength is not different. So that would mean that there's no reason in terms of strength measures for elderly men to start changing their movements. Still, when we observed them, we saw that in 72% of the men, or 72 trials of the men in normal velocity, so at the normal speed, they start using their armrest, and the elderly men in the fast speed actually had 89% of the elderly men used their armrest. But I have to say that 100% of the men in all 100% of the trials, the elderly men used a way of pushing off. So the other men used either pushing off from the thighs or used an asymmetric push off. You could say, well, is that bad? Well, we saw this is a compensation strategy, and compensation strategies can be detrimental on the long term. So imagine that as soon as you start using pushing off on your armrest, that means that you're not using your lower limb muscles that much anymore. So that could mean that there's an accelerated decline of your lower limb muscle mass. And of course, there are other compensation strategies that are interesting that might be detrimental on the long term. So that's why we wonder what moves us. Why do we then start to change into a different strategy in daily life activities? And we have two terms for this. One was our idea, well, we could compensate for capacity. So indeed, if our strength is reducing, then maybe we have to change our strategies. But what we saw now is that the capacity of these elderly men in strength is definitely enough to do the same strategies as the young women. So there must be something else in play. So either that could be in one of the other capacities, like neurocapacity, which from the results so far, it doesn't look like that. But there's something else going on, and that's compensation for movement objectives. So when we look at walking, if you walked out this door, when we looked at the speed skater, it was really clear, right? They want to go as fast as possible. So the velocity, their aim was to go to reach that finish line as fast as possible. But when you walked out the door this morning, if you left your house today, by the way, you didn't really think about what velocity you wanted to walk at. And we saw from previous studies and from also animal studies that you usually choose a velocity where your energy expenditure per unit distance is as low as possible. So we're quite good optimizers. But what we know is that elderly people, that everything, so the whole movement will cost more energy for these people when we age. However, we also see that their velocity is lower. So either they start to optimize, which is something that's not really expected, or there are other things at play. And these other things could be avoidance of pain, or it could be that they're afraid of falling, so they put more emphasis on stability. But you can imagine it's quite difficult for, if you want to we need to understand this, if we want to capture the boundaries of human movement. But it's very difficult to do this experimentally. And that's why we started to make models again. So we want to combine the experimental data with predictive models. Because what we can do in a model, like a tower of Jenga, we can basically take out single blocks one by one and see what this eventually does to our movement patterns. So in London, we started on a lower a self-learning model. So I just show you in the on the slide here how it sort of learns. So what it gets is it gets a certain capacity. So we build a neuromuscular model. We can put a certain decline into this capacity. We can add the movement objectives, give different weighting to these movement objectives, and then the model with this certain structure tries to complete a certain task. So where we usually use inverse dynamics, where we say this is the kinematics. What are the forces? So how did we get to these kinematics? We're here saying we're not giving you any kinematics, we're just giving you a task. So you can use any compensation strategy that you want. What would be the compensation strategy that you would use? Well, the lower limb model for this, for sitting to walking, is done, and we've worked on this. We've tested a lot of two different foot strategies already, different foot seating heights, but there's definitely something missing here, because we know that a lot of the participants actually used upper limb compensation. So to move this further, the next step is basically we're building this into a full body model where we can also look at the compensation strategies of compensating by using your upper limb. And together with these models, using his models, basically, we need to know what moves us, but also with these models is the only way to find out what moves us. And this is very important if you want to know the boundaries of human movement. We need to know how we optimize, because otherwise we will never be able to find either the upper bounds or the lower bounds of human movements. So that's what I wanted to tell you guys today. Thank you for listening.
Fantastic. Thank you very much. And once again, I offer you a round of applause on behalf of every other person in the room who I'm sure is applauding at home. I can see Anthony definitely is. And once again, we have a question already. If you do have a question, stick your hand up and you can ask it directly, or stick it in the chat and I'll read it out, whatever works for you. So Tanel Laurent asks, what was the method used to measure the muscular capacity of your test people?
Yeah, we've done two different things. Or three actually. So the first one is on a Cybex machine, so a dynamometer, we measured the upper and lower limb maximum joint moments. So maximum joint moments. On the other hand, we also did the nerve conduction study, and from that we could also see the maximum M wave, so Mmax. And we did a hand grip strength, which is I know it just seems only the hand grip, but it's a clinical test that's used a lot to give an estimate of the overall frailty usually, but that's something we incorporated as well.
Fantastic. So just following on from that question, I was interested. So you mentioned that the elderly men and the young women had approximately the same strength. Did you compensate for the mass they had to move? Like if the elderly men are fatter or slower, then presumably they have to apply more force in order to achieve the same goal.
Yeah, absolutely. I forgot to say. So this is all compensated for body mass.
Right. Okay.
Yeah.
Fantastic. So Puya Amiri asks, what was the most important factor whose decline for the elderly caused the most significant changes in the sit and stand task of the elderly according to your forward simulation?
Yeah, so that's a question I can't answer yet based on the forward simulation. Interestingly enough, but we don't really have a reason for this yet, is that the biggest change we saw in the measurements is actually joint range of motion, and especially hip range of motion. So we still, and that's something we're trying to find out now. It's with the predictive modeling as well, why that would be such an important factor.
Cool. Thank you very much. We've got questions rolling in. So Jared Antflick asks, at what sampling rate, how many hertz did your undershoe force plates capture data at?
The instrumented skates, I think, so we measured it at 100 hertz, but they could go up to 200 to 500 hertz even if you want. But yeah.
That's great. So is there an advantage to going to 100 hertz, or is there?
So for us, it was because we had the motion capture data at the same sample rate, basically. And it also depends on if you want to do it real time. So if you want to get your data real time, you have to go to a slower rate. But we could also capture it. We could save the data on the data logger, and then you can go up to 500 hertz.
Absolutely fantastic. Thank you very much. So Naomi Nakayama asks, the way we move our body in our brain, does our memory of movement affect it?
Yeah, that's another thing we want to look at. So especially because what we see is if in patients when they had an injury, they can keep a certain compensation strategy for a very long time. So the question is, at some point you seem to stick to a certain strategy, and then the question is, do you go back to, can we move someone back to their old strategies, and how did they get to this particular strategy?
Great. And we have another question from Puya Amiri, who's very prolific today. How do you model pain in your objective? How's that metric assigned in your model?
So we have pain at the moment. It's one of the movement objectives. So what we do now is we're trying to unload a certain joint. So if you're saying, well, I have pain in my right knee, if my model should simulate this, it's unloading that knee as much as possible. You can also say, you can even give it a wave factor if you want. So you say it's okay if it is this percentage of the other knee, for instance.
Okay, and do the exact weightings make a big difference, or like presumably you need to like small changes might produce a very strong effect in your model, and how accurate do you think that is?
Yeah, we said so. The thing is with these models, it's you need to think of this always relative. So there's never a so even if we put energy in there, of course the energy measure is never the same as an actual energy measure that we can't simulate that yet in humans because we don't know how to do that yet. So it's always a relative measure. So it's as soon as you introduce pain, you basically can see that there's a relative decline. So it doesn't really matter whether an 8 in my model is the same as an 8 in an experiment at the moment.
Great. Okay. We have a question from Neophytos Viralides. I do apologize. I've just butchered your name. How were these models of the lower limb designed to self-learn how to find their balance in walking? I realize that's a very big question. So
yeah, because I'm thinking, what kind of, so on the one hand, so it's learning. So the learning of that is a CMA optimizer, by the way. It's finding their balance because we have a balance basin. That's not the word. It's the stability margin. Sorry, that's the English word for that. So we incorporate the stability margin and it has to stay within a certain margin of that. So basically it's saying, if I would get perturbated by a certain force, will you still be upright? And how big is that force that I can perturbate you with?
Great. Okay, so very concise and to the point. Thank you very much. We have time for one more question if anyone else would like to jump in. Oh, yeah. Oh, no, sorry. That's just a thank you. Would anybody else like to jump in at the end? Nothing so far. No hands raised. Well, in which case, I will finish right at the end. You mentioned your speed skater model had a 3% error margin. What would you say the difference is, or the required difference is for an athlete? Like, does an athlete need to be 1% better in order to win a race, or 10% better? Like, what's the margin for success?
Yeah, it's even way less than that. So it's they estimated something. It's 0.01%. But the problem is the model might even not be the problem, but the measurement systems that we used are less accurate than the accuracy we get from the model at the moment. So having a motion capture system is in this huge volume doesn't go higher than 1 centimeter accuracy, and that means you get so. To give you an idea, if you have 1 degree of difference in your skate, you get or in the orientation of your skate, you get a 10% difference almost in your power estimation. So it goes really quickly. So at the moment we come, and this also accounts that once someone calculated this once, this even goes for the systems that are used in the actual matches. So there's been a difference in a match where someone was sitting on the inside of the rink and the outside of the rink, and the difference between these two skaters was so small that it was smaller than the difference of the sound of the pistol going off and actually having to reach the other person. So there was especially in the 500 meters at the moment, they're getting a problem with measurement systems in the games themselves.
Great. So it's not just us. It's affects everyone. Great.
Yeah.
Well, in which case, I'm afraid we've overrun by a little bit, but it's an ideal opportunity to thank you and both Andy. I don't know if Andy's still here. There will be an opportunity for people to stick around to discuss the topics that we've discussed today. I'm afraid I have to run off, but if you want to stick around and have a chat, unmute yourself and socialize with everyone else that joined us today, you're more than welcome. And I don't know if Elina will be able to stick around for a little while longer.
I can stick around for a bit.
In which case, I will offer you a round of applause on behalf of everyone else. So, excellent job, both to you and Andy. And let's close the session.